to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 18th episode of the Weekly Bioanalysis. My name is John Perkins. I am the Senior Scientific Advisor for all things related to LC-MS technologies. I've been with KCAS for 10 months, but I've been in bioanalytical for over 25 years. I'm here with my co-host, Dominic Guarino.
Hello, my name is Dominic Guarino, and I'm a Senior Scientific Advisor for all things related to ligand binding assays, biomarkers, and flow cytometry. I've been with KCS for about 7.5 years now, and I've been in the bioanalytical space for close to 26 years.
Today we have a special guest. I'd like to introduce Dr. Amy Mize.
Hello, this is Amy, and I'm happy to be here today.
Today's podcast, as always, is brought to you by KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical laboratory located in Kansas City, Kansas, serving the pharmaceutical and biotech industries for over 40 years. Dom and I are senior scientific advisors at KCAS, and either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS's services. We are thrilled to have you listening again. As mentioned, this will be our 18th week recording the podcast, so if you're interested, the other episodes can be found at kcsbio.com. As a reminder, you can also follow us on Twitter at @TheWeeklyBio and at KCASBio. As we mentioned before, we started this whole thing because of social distancing getting in the way of normal communication. Even today, Dom and I are in completely different places. I'm in upstate New York, Dom is in Kansas City, our guest is in Tulsa, and our producer is in a different part of the Midwest. This weekly podcast gives the chance to connect with all of you, and we hope it is a chance for you to get to know us too. Even more, it's a chance for you to get to know KCS and the services we provide. As always, a review of the latest news and resources, and then a focus on the topic of our choosing before discussing any feedback we've had from you. We're always looking for new topics, and we'd be happy to discuss something that you want us to cover. Then we'll wrap up and give you a little teaser for next week, letting you know what is coming in the future. So again, we're thrilled to have you here, and we're looking forward to a fun episode today. To kick us off, Dom is going to go over today's agenda and podcast topic a bit before we jump into the news and resources section. Dom.
Yeah, thanks, John. So if you're unfamiliar with our podcast, the first portion will go over some news and resources, and we typically like to talk about the latest on COVID, go over a little and then go over some non-COVID related news and resources. And then we'll talk about our main portion of the podcast, and today we'll be discussing dose form. We'll talk about what is dose form, why it's important to BioA studies, what are some of the challenges, and then we'll touch on what life is like for a CRO in the dose form space. So we'll move on to some feedback and questions, and then we'll do a little bit of a teaser for next week's podcast and talk about a topic of our choosing. With that, John, let's jump right into the news and notes. So the first is the outbreaks on college campuses. I think we just wanted to touch on that. And surprise, surprise, you get a bunch of adolescents or teenagers, late teens, early 20s together, and they're going to go together with greater than groups of 10. Although I have heard of one university that is smaller in size that's not allowing more than 10. And if you're caught in a situation where you have more than 10, they actually will send you home. And that's happened to like 110 kids on the campus or something. So, John, just let's quickly, I know North Carolina is one school, but what are your thoughts on colleges?
Well,
I don't think anybody's surprised.
For us, we're actually just at the beginning of this. Cornell is our biggest university close by, and they had students coming this week. There's a open air testing facility by one of the malls, and so that's where students are directed to go for testing. So it's probably too early in this in the whole process of things to see how Cornell is affected. But having spent 3 years in North Carolina, it's always interesting to watch the how things are happening in North Carolina compared to here. I was reading an article last night that NC State has called the keeping of students in halls of residence untenable. So they're obviously struggling to deal with it. They've had a number of hotspots in Raleigh, and so they're trying to figure out what to do with that. They're moving to online classes, which we're seeing a lot with the Carolina schools. I know Eastern Carolina University has gone fully online as well. So
Yeah.
it's just a huge challenge, and I mean it's a logistical nightmare, to be honest.
Yeah, well, locally, Missouri has, after I don't know, it was less than 5 days, Missouri University has said, "Oh, we're all online." And really, this is just we're not going to go down a path of how wrong that might be to get someone to commit to 12 or 8 months worth of housing and then tell them they can't go anywhere. And then KU University had an outbreak, but they're still going, I think. And then K-State had an outbreak, but the numbers weren't as big as Missouri. So time will tell. Let's move on to the next subject, but I think it was interesting to touch on.
Funnily enough, my son is actually enrolled at K-State, but his course is actually fully geared to be online, so there's no need for him to visit campus at all, though at some point we might take him over there. Anyway,
you could come to it's in Manhattan, Kansas. They call it the Little Big Apple, I guess, and it's a nice place. So, John, interesting on the next topic is the Biogen is not putatively, I think it's been proven that the conference they had at the Long Wharf Marriott in Boston was the source of 20,000 cases, although I had initially thought it was deaths, which made me, a morbidity bigger, badder, but still it's mind-blowing to think of how 20,000 cases is stemming from a room of, what was it like? I don't know, 30 participants or something. So
yeah, I think it's interesting. Basically, they looked at the sequence of specific COVID-19 mutations, and this team who did the study, and based on those mutations, were then able to identify a number of people that had the mutations that could be traced back to this conference. And although their sample size was small, they extrapolate to the 20,000, but still, yeah, it's incredible how a small gathering, how far reaching this can become with time in terms of contact, etc. Which actually takes us into the next topic, this new recommendation of the CDC that asymptomatic people don't need testing, which seems really, really backward. I mean, to me, they're exactly the people that you do need to test, because if you're asymptomatic, you don't know you have anything wrong with you, you're going to be going out in public and doing things as normal, and you could be affecting any number of people with no knowledge and really no fault because you're not aware. The testing of asymptomatic people, and it's a 40% population, really, I think, is absolutely vital.
Yeah, I think it caught us both off guard a little bit. I don't know if there was sometimes I wonder if it was misreported or misunderstood, and they're going to walk it back, but because it happened so recently. But John, it is really kind of hard to imagine how grandma and grandpa are feeling about asymptomatic. If you're over the age of 70, right, then you really would want to know who you're coming in contact with. And I don't know, it really, I mean, I kind of get it, but maybe it's just too soon. I don't know, but it was a really weird sort of thing that made CNN news, not just our kind of more nerdy news, right?
Yeah, I mean, I think there's definitely a lot of concern in the medical community about this. I mean, certainly symptomatic, you get tested, but really you need to test all the asymptomatic. Is a case of just knowing how far it is out there, and to just say these people don't need to be tested. I think you're sitting on a powder keg if you don't trace the asymptomatic people.
Yeah, we'll move on, but it was definitely just a really bizarre. I don't want to call it necessarily shocking, but you just don't need to say anything like that. I think it was just a weird timing. So the next one, John, I thought this one was a little bit. I hope we get ahead of it. This is around nursing homes. They say majority of nursing homes won't last another year under the current conditions, and so I think what that means is they say 72% of the responders said they wouldn't make it another year under the status it's at, right? So it comes around profit margins and things like that, but really it has to do probably more with the whole social distancing, and then how even on a sidebar here, I had a sleep study done this week to get a CPAC. I'll open up, and so it was so hard for them to come in and out of the room. I can't imagine what it's like if on a day-to-day by basis, every time somebody has to come in to see somebody who is potentially positive or to cut down risk, has to gown up why they're struggling. I think that's the
Oh,
root cause. It doesn't really, to me, it doesn't touch on it in this article, but in my brief experience, and it wasn't necessarily a, a, um, a, um, a um, nursing home setting. However, it had nurses that were there during a home setting, and I could see the frustration in them alone because they had to like put on a whole not a bunny suit, but they have to like gown up when they come in, and then they got a degown, and
yes,
that's
I mean,
cost. That's a big
every,
hurdle, not just time, but that
yeah,
gown costs money every
yeah,
time too.
everything slows up, and then you say they look at the, supplemental pay effects things, the increase in cleaning supplies, because again, wherever you go, it's like, I mean, when I get a haircut, there's additional
Yep.
cleaning steps between people sitting in the chair and stuff like that, which all adds to your not your bottom line, but your costs that affect your bottom line. Yeah,
No, that's a good point about the barber shops and things like that too. Yeah, but no, we're experiencing that. It is. I guess it gets passed on to the consumer. Maybe they got to figure out how to do it on the nursing home side. But I think it's good to talk about it. And they still say another year, so maybe something. Let's hope. Let's hope that all gets straightened out, and something we'll try to maybe circle back on in 6 months.
sure,
But
yeah, absolutely.
interesting read, and they're not. It's not going away for the foreseeable future. So. Next one is, and then we're doing pretty well on time, John. The next one is the GSK vaccine awareness. So this is a campaign to not just COVID-19 specific, but vaccines in general. And it was some surprising numbers. I'm continued to surprised to see at how reluctant people are to take vaccines. 72% surveyed realized how important they were, but only 47% said they'd take it, or
Yeah,
uh,
it's
2%.
also
But
awareness
it just
of
cracked me up. Sorry, go ahead, John.
So it's also awareness of other diseases that can affect, particularly older population. I think it was people not being aware of pneumonia vaccines, shingles vaccines, things like that. And so this GSK campaign is not only to increase the take up of vaccinations, but also to make people aware of what are out there to help you stay healthy.
Yeah,
And I think it's definitely a good and timely thing to do. I
it's
mean,
shocking, John. 1 in 4 elderly have not heard of the pneumonial vaccine, the shingles vaccine, and the tetanus or the Tdap,
yeah,
tetanus, diphtheria, and pertussin. Which I think that one I'm even getting a boost on next week. They say, or next 2 months from now. That one they recommend you take every 10 years now.
yes,
But I mean,
I'm
I
so
don't know, John. I'm taking every vaccine imaginable. I don't care if it gives me slight infection. Don't care.
I'm so accident prone. I'm pretty regular on tetanus with the top up.
Yeah. Oh, of course. My silly luck. I refused one on a wellness visit because I was rescheduled like 90 days later to do something else. And sure enough, I'm out there like moving boards with nails in it. I'm like, oh, I should have had it done. Thankfully, I did not. This was part of my deck. Anyhow, John, let's move on with the news there.
But
Let's,
a positive on the vaccine front, yeah. Let's skip the other topic. But let's move to Africa,
yeah.
which this is a
Your
huge.
favorite continent, by the way, John. You have such good knowledge of it, and you're such a worldly guy. I just have to throw that in. Podcast number 18.
Yeah, I think this might have been. Yeah, it was actually someone I follow on Twitter actually highlighted this, and yeah, there's all these different sources to pick up this stuff. But I mean, this is an incredible story that basically on Tuesday of this week, Africa declared itself free of wild polio. There's still the issue that there's actually polio vaccine related cases, which are a very very small proportion, but effectively there's been no wild cases in Africa for 4 years, so
Yeah,
they've declared the continent free. And 25 years ago, thousands of children would be affected by this every year, and it's gone. Yeah, now we're the only countries, according to the articles that we read, is wild type or wild. Yeah, wild polio is only in Afghanistan and Pakistan, but this is really this is a huge endorsement of the importance of vaccination and what it means to the quality of life.
Absolutely, John. And even those disease, vaccine related ones are way more mild if there's a milder form of polio than the wild type. So
And
just
and the,
wonderful news, and really um,
and the other part of
makes you really a little more optimistic towards COVID in general when you read those types of stories.
Exactly, and another part of when you say the vaccine-related cases are, mild, they're also ready to deal with them, and so it's not it isn't the same tax on the medical system. And actually, if I think one of the two articles was saying they're actually applying the lessons they've learned from polio to how they're African countries to how they're treating and monitoring COVID. So they've learned a lot. It's a huge initiative across the continent, and really is a huge triumph for all those countries.
Yeah, and that's an interesting. This is completely off topic a little bit, but I was reading more about the 2003 COVID or SARS or MERS outbreak, and there was more deaths related around there. I forget the number. I wanted to go back and look. I feel like it was like 800 or something died, which was surprising. But then what my point there is, the vaccines are further along because of that. So
Mm-hmm.
I'm getting more in of the belief that the reason why a lot of these vaccine trials are accelerated, we've talked about a variety of things, but it's also they had a good building block from 2003 where there were labs that have been doing it the whole time that are now, most notably, even Pittsburgh has been doing it. That was part of what I was reading around my alumni there. So anyhow, John,
Yeah,
great story.
I'll just
Love
say
to
one
hear
more
about
thing.
vaccines
I just
working worldwide. Go ahead.
I'll just say one more thing on that. It's interesting you say about learning from SARS and MERS. I think a lot of the companies who are involved in looking for not just vaccines but other therapies are all in this. A lot of them were in this field anyway, and have learned from dealing with Ebola, SARS, MERS, and various other viruses, and
Even
applying
dengue, the
that
recent dengue
modeling.
virus, the companies that were working at that because like you said, there's whatever worked in Africa around the polio, they're going to have to get and recapitulate it that way. So great stuff, John.
Yep.
We could spend a whole another hour probably talking about vaccines and just anyhow, let's move on to our let's make it the last topic. Um, let's go with we're going to stay apolitical and not talk about the Trump pressure, but let's do it anyways. FDA faces a reputation crisis amid Trump pressure for fast COVID action. And that's bad news for pharma. I wanted to point this one out, John, because um, we had talked about some of the momentum in the pharma space beforehand in terms of perception, right? And
hmm
so there had been a good feel about pharma, and then this kind of comes out. And I don't, to me, it's, you can spin it one way or the other, but I don't know how much I like any of these sorts of. When I say spin it, you could say the opposite. It's look
I,
how good it's doing, right?
I
Um,
think my take on something like this is I think you always have to be aware of what the perception might be of some of these things. I mean, I know as a customer service organization, it's you'll get say for example negative feedback from a customer, and you're looking at it and think, but I didn't do anything wrong here, but ultimately it's their perception, and that's the reality you have to deal with. And I think that's the issue here. If this is perceived that the FDA is losing independence, then that becomes the reality that they have to deal with. So it could be very it could be a very negative thing. But yeah, I think it's just something to watch.
yeah, and well, but it is, and then the former FDA global there, Gobble is how you say it, it's not Gobble.
Is it got?
Um, he makes a motion, and then David Kessler, who's again another former FDA, is on the Biden presidential side. So then that stuff like that just clouds the picture, and really we
Absolutely.
definitely don't want anyone influencing or putting pressure on the FDA like unjustifiably, with or at least without some resources and things of that nature. If you're going to say make it quicker, then staff the FDA to do it. So anyhow, it's just we'll
That's
keep
the
that drama.
important thing. It's give them the resources to make the right decisions, knowing that we all want this to be accelerated, but it has to be done the right way. And
Yeah,
and like you
pounding your
said, that
fist on the table and saying, "Oh, we're going to get it done by January," is not that's not how it works, right? Um,
absolutely. Yep.
so let's. I think we can. There was quite a bit of movement in the IPO space but we don't have to drill into too many details, John. It's just there's lots of funding out there, and it's safe to say I think the drug development space is continuing to start to loosen the purse string slightly. You see clinical labs are starting to rebound slightly, but I do think one thing is supply chain is becoming a challenge, but maybe that's
Sure.
something we'll deal with as part of our next week's show.
Yeah, we won't maybe touch in the future, but I just saw like another fierce biotech today talking about the Pfizer BioNTech vaccine. They're getting very concerned about potential impact of supply chain on that vaccine, even though a couple of weeks ago we were talking about that one as the potential front runner.
Yeah,
So yes, it's really important.
we got to
So
repurpose all these empty office spaces because everybody's working at home and make them into factories.
there you go. Yep, you've
And
solved
all the
it.
restaurants that people can't go to or aren't working in, they're going to turn into making glass vials for vaccines. But I kid, but on the same, I did read it was someone was hiring, Regeneron's hiring like 800 people. So that to help with, and I didn't quite read where that is, but that is happening. And, that's just, it's part of progress, I'm guessing. But anyhow, let's move on to our main portion, and what an exciting main portion we have. We have a guest visitor, a guest co-host. We love that, John, right?
Yeah,
Her name is Dr. Amy Mize. She is the Vice President of Business Development and Marketing here at KCS. She has been in the CRO space for quite some time, but we'll let her share a little bit of her story. We're going to be talking about dose form and let's just say the what, why, challenges, and what to look for in a CRO are some of the topics. So
yeah, just yeah. We talk about bioanalysis most of the time, but as we've also acknowledged in previous podcasts, there are other pieces of the analytical slash supply chain that is critical to actually support the bioanalytical data, and that's the support of bioanalytical data is where dose form analysis comes in. I'm not going to say too much more because I really want Amy to say it, but we thought it was important to focus on this service because it fundamentally underpins much of the data in non-clinical studies. Would you like to introduce yourself, Amy, and give us a bit about your background, and then we'll take it from there?
Sure. So, certainly excited to be able to join the podcast today. It's my first opportunity to be part of this program. So, as the two mentioned, my name's Amy Mize, and I'm the Vice President of Business Development at KCAS. And I've been with the company going on 3 years. And prior to my time at KCAS, I've been in the CRO space for just about 20 years, both in the operational aspects and in the business development realm. Most of my time has been in the bioanalytical dose solution, both non-clinical and clinical space, as well as overseeing in the GMP area. So really in all aspects of the drug development pipeline in the CRO space. So that's really what my background entails. And obviously today we're here to share a little bit more about the dose solution space.
Let's start with just a simple definition of what is dose form and what is involved in dose form analysis.
Yeah, so I think it is obviously a pretty simplistic in its thinking, but it actually can be quite complicated when you get in the laboratory to work on the testing. So it really is mostly part of a non-clinical study in the support of the toxicology support. So in its simplest thinking, it really is around the concentration verification of the dosages that are used in the toxicology study to verify their content and their concentration. So it can be just the concentration verification. Sometimes it goes into understanding the excipients or the different components of that dosage and what the counterparts of that dose form. The other parts that are typically tested as part of a dose form study can be things such as the homogeneity, depending on what type of dose form it is. There's obviously lots of different types, from just a simple solution to a suspension type of dose form. So really depends on what type of dose solution it is. But homogeneity is a typical type of test that is part of a dose solution study. Obviously, stability is a typical portion of that kind of test. And then compatibility is another typical component of a typical dose form study. So there's a lot of components, and we can get into a lot of other detail of it. But in its most simplistic basis, the dose form study is to test for the concentration of the API or the active pharmaceutical ingredient in the dosage that's going to be given as part of a non-clinical study.
So, actually, one of the terms that you mentioned there, I haven't heard before. What's the compatibility?
Yeah, I was jumping on that one too, Amy. You've struck a chord with this audience now.
Yeah, so the compatibility is really around the container that the dose form is going to be placed in. So let's think of maybe a more difficult type of study where let's think of a monkey study, for example, where maybe the dose is going to be in a kind of syringe, and they're going to have to use a variety of different tubing to be able to administer over a time course. Let's assume it's not a simple, injection or even an oral gavage. Let's assume it's over a period of time where they're infusing. And so the infusion has to go through a series of tubing. And then let's assume this drug is sticky and is going to adhere to not only that syringe, but also the tubing. So the compatibility study is around all the aspects that the dosage is going to touch throughout the syringe, the tubing, all the different parts between where it's injected in the syringe and then all the components it touches before it gets into the animal. And so you have to, in essence, test all the different parts and pieces, the syringe, the tubing, wash that out and look to see, is the dose in essence stuck or sticking to any of those parts? Is it there, or did 100% of that dose reach the animal? So the compatibility study is set up to look at all of those pieces and parts. So at that point, you really have to decide and understand how the dose is going to be given. And there's a lot of work that has to be done between the analytical testing facility and the in-life facility to really understand the non-clinical protocol and how that study is going to be handled. So those studies are going to be the more complex types of studies. That's not going to be a study that's necessary if the dose is, oral gavage necessarily to a rat, for example. But the compatibility study is more around the container and the storage container maybe, or even the dosing vehicle and the dosing container that the dose is going to touch between it being prepared and being dosed to the animal on the day of dose.
Yeah. I think for a bioanalytical person, it's easy to underestimate how complex dose form analysis is. I mean, I sort of know from my perspective, we never really did it, but you knew it was using LC with UV detection, whereas we're thinking LCMS, and the complexity for us is on the mass spectrometer, and you sort of almost gloss over some of the challenges that you're dealing with. I mean, such as like you're talking the potential absorption issues, and then obviously for us doing sample analysis, we're looking at drugs that are solubilized in liquids, but here you're dealing with suspension, so that making sure you got the uniformity for sampling and testing from there. I didn't really think about that until I start, talking to you and David Masters more about this, and I'm glad I'm not doing it.
Yeah, well, I want to kind of shine the other lens as we normally do. So I'll jump in the on the biologics side. It's for me, I didn't know or focus too much on it until you come full face with accusations around you're not doing this right because the levels aren't correct. And then what the dose form is a great area to do some troubleshooting in the LBA side. And what I mean is oftentimes levels can be reduced below and they were never actually put into the animal because of some of the things Amy was touching on. And it's probably a higher frequency to have sticky things that are over a long dosing time or dosed at sounds a lot like a biologic. So
Sure.
certainly, it's something that I didn't know the term. I never knew how complex it could be based off of what Amy said, but I have felt the wrath of a laboratory that's done a poor job understanding that. And on our end being like, we don't know what to tell you. And then a month later, someone comes back and says, hey, test this sample, or we have measured some dose form, and it's become a bigger part of our process here at KCS since David's joined a couple years ago now. So, Amy, let's get back to you. I
Yeah,
think you did a great job of going over some of the whys it's important. You even touched on some of the challenges, but why don't you share whatever you like to talk about next in terms of dose form?
yeah, I would just say the other pitfall, we've kind of touched on it a little bit, but if you compare and contrast to things someone would think about getting their bioanalytical method set up, and maybe you don't think about in getting your non-clinical method set up, and then you pitfall, and then it bites you in the end of the study. So you're constantly thinking about getting your bioanalytical method set up ahead of time. You work to develop that method and get your method validated oftentimes well ahead of sample collection, because you want to make sure you're collecting the sample in the proper tube and you're storing that bioanalytical sample at the proper temperature. All of those things obviously very important to, the status of your study. Well, the same thing holds true for a dose solution sample, but oftentimes it's not thought about. So you get into the study and now you haven't set up your dose form method. And now you've dosed your animals, but you don't have a method to test the concentration of those dose form samples. So now your dose form samples are sitting around for days or weeks, and the lab now has to set up a method. So I think we oftentimes see sponsors forget that there is also complexity in setting up that dose form method, just very similar to the complexity in a different way, setting up that bioanalytical method. It takes time. It takes days, and you have to not only develop that dose form method, you have to validate that method, and you need to do that ahead of time so that you can understand the types of containers the dose form can and can't be in. How much stability do you have? Is it hours? Is it days? So that you can make sure you're doing all the right things for your study. And if you aren't ahead of that and you don't do that properly, then you can just as well compromise your study for the dose form samples, just like you could for the bioanalytical samples. So I think that is something we've seen at times where a lab in a sponsor isn't as organized, and then dose form samples sit in the freezer too long, and then we finally have the method ready and we analyze, and now that dose form sample comes back at a concentration much lower than what it was expected. So now, of course, investigations have to be opened, and the question is it because the sample sat too long, or is it because the animal was dosed with a lower concentration than was expected? So that can open lots of questions that aren't necessarily fun ones to answer in the middle of trying to get an IND filed. So I think that's just another pitfall in forgetting how complex these dose form analyses and methods can be, and trying to be prepared for it and make sure those methods get set up ahead of time, just like your bioanalytical method needs to be set up ahead of time as well.
Yeah, Amy, there's so many more questions I have around what you were discussing just there because of not just for me on the biologic side. I think this is part of what you just said is why doing um, a non-tox study. The word is escaping me. I think it's like just like an investigation or pilot study before you do your true tox. And we, as both John and I know, and Amy, labs that invest in that are always the ones that tend to have a much easier and less investigations. That's what that translates into in the bioanalytical realm of things. When they make the assurances that any of the kinks are ironed out before you get into those IND supporting trials.
Absolutely. And I think if somebody asked me, what is one of the main challenges in just a dose form study, I honestly, one of the basic challenges I think is finding a suitable lab. So I think the three different areas are: you either find a tox lab that is very good at the in life, but maybe not as well set up to do the analytical as scenario one. You find a bioanalytical lab that's very good at doing bioanalytical, but doesn't necessarily understand the strengths and differences between a bioanalytical sample and a dose form sample. Or you find a GMP laboratory that obviously understands API and dose form, but they are GMP laboratory and don't understand the differences between doing work in a GMP, getting things ready for in a clinical sense, versus a non-clinical GLP study and just the differences in those regulations. And there are some differences there. So those are the three scenarios we see most often. And none of those are the absolute best choice for this kind of study. And so finding a laboratory that's really skilled and understands and is really set up to do your GLP non-clinical dose solution testing and understands the differences and what it takes to develop a method, validate that method, and be prepared to run your dose solution samples in a timely manner to be prepared for your study is really a challenge and something I think you really have to look for, be prepared for, and to be successful for your non-clinical program in total.
Yeah, I think from my previous existence, we had a dose form group for a short while, and it was certainly recognised that you need that standalone team who are focused on it. But ultimately, we still came at it from a bioanalytical direction, so didn't I think appreciate all the complexities that you've outlined here, and longer term, the group we moved away from dose form analysis. What about the technologies that we tend to use?
Yeah, I think the most common, there's obviously, depending on the molecule, that's obviously going to drive the technology that's the best choice for the molecule. But I would say the most common technology that's the best choice, 9 times out of 10 for dose form analysis, is a standalone HPLC method with typically UV detection. And that's typically because the very high concentration of these dose form samples. And so that's just the best, analytical technique of choice. Now, that might not work for some of the biologics, obviously. So it really just depends there. But I would say most of the time, we tend to use HPLC UV as our technique of choice. There are times where going with mass spec might be the way to go. And then there's obviously times where we're even doing dose form work using MSD or some other plate reader technology if necessary. But I think the real pros and cons of going with an HPLC UV or fluorescence technology is because of the high concentration in the dose solution sample and the fact that you really want to have as tight of a criteria in the accuracy and precision of that method as you can. So you're really trying to limit the variability in the methodology. You don't typically use an internal standard where a mass spec method is typically going to need the use of an internal standard, and that's just atypical for a dose solution method. So typically that's going to be the analytical choice that we're going to go with as our standard for most dose solution methodologies.
Look at this. I can't believe how much I'm learning about my own organization on today's podcast. I had no idea you didn't need an internal standard for dose form. Just blows the mind. It's awesome. Well, Amy, I think we've covered a good portion of the agenda. We certainly can discuss more if needed, but this has been a wonderful, at least for on my side of things, a wonderful break from the norm to have you on. You kind of touched on a couple things that might warrant you having to come back. I feel like there's some sort of discussion around how a GLP laboratory with good understanding of GMP gives us a competitive edge, because that's kind of what you touched on. And so maybe we got to bring you back. I'm sure.
And certainly, just as an addition, there is that where we talked about the different technologies, and we touched on this in previous podcasts. Having all these teams under one roof does give us the ability to jump between different technologies for dose form, just the same way as we would do for bioanalytical. It's that flexibility of technologies is key to sort of our ability to support this as well.
Absolutely.
Yeah, and there's a lot, that you said. It was new to me, and I think there will be a lot of our team members that will be very happy you joined us to kind of shed some light on dose form. And maybe next time we should even bring David with you, and we can kind of tackle it at even a more practical level, and maybe even some KCS case studies. But I will share on my side of things. I can't begin to let you know how important it is to have a good dose form method ironed out, because I can think of a couple instances where it really was something that caused quite a bit of, let's say, loss of time and money for two of our clients And what the root cause was that in fact they did not dose right.
Yeah, I
And it
mean,
happens. It
it,
does happen. So
yeah,
anyhow,
it is important because a lot of the time there's been, preliminary studies that, the customers come to us has an idea of what the drug is going to be doing in the animal or whatever. And when these numbers don't come out right, that causes a lot of problems. And like you say, the investigation and headaches to fix what's happened. So yeah, it really is critical to have a dose form right.
the good news is there's been some movement on the biologic side that people that are doing bispecifics or multivalency compounds or what they call Franken compounds or chimerics, they are now tackling dose form upfront because in the past it had kind of been you know, I don't want to say a throw in after the fact, but it was kind of a throw in after the fact. And from a bioanalytical standpoint, it was brutal. So those companies in that space are getting more ahead of it, and regulations are coming out on how to handle it. So I hope that made sense. Those are the sticky ones, and those are ones that Amy specifically mentioned can be problematic. Um, well, with that, I think we'll say thank you. Amy, any last departing words or any words of wisdom for our audience?
No, thank you so much for letting me join today, and I'd be happy to come back and answer any follow-up questions that
Well,
viewers might have.
but you're not done yet. Just so you know, when we
Don't
start
go
to talk
anywhere
about our
yet.
weekend, we're going to ask you some more questions. So don't go anywhere.
I won't.
You're not off the hook all the way. So I don't know if we have any feedback. That's what
We
we're at
we
today.
we have limited feedback, but let's move to the feedback
Oh, I
section.
see it now.
Basically, we had a note from Rachel who said, "I had no idea I had missed a few episodes lately. I'm looking forward to catching up. Well, hopefully you'll enjoy what we've been talking about in the interim, and we're still going to keep doing this. So we'll hopefully have a healthy supply in the future for you too."
Yeah, well, I have not the bolus of episodes. I have not gotten caught up myself, so this is great stuff for me to do over the weekend. I am going to listen to them again, John, because we can't get better without doing those types of practices. But thanks, Rachel, for catching up. And I've had a few feedback from just some clients and internal. They had also noticed that there was a slight hiccup with the episodes, and I've been asking people to provide feedback and topics and things of that nature. But we did get some notice from, I don't know, on my end, at least 4 or 5 people shot me some sort of message around, oh, that's where they all had been, which made me feel good, John. I don't know about you. I like anytime to know it's somebody's at least listening. So, topic for next week, John, are you in agreement that we'll do sort of our QBR or a quarterly business
Sure.
review of drug
Yeah.
development during a pandemic?
Yeah, that sounds good. I'm not quite sure what it refers to, but yeah, let's go for it.
Yeah, well, I mean, I think
We've
we
got some time
initially,
to figure
if
it
you
out.
if you think about a few, maybe episode 4 or 5, we were very heavy on supply chains and what was happening in the market. So it's going to focus a little bit on that, and then maybe even share a little bit around what we've been doing COVID wise.
Yeah, sounds
That's what
good.
that means. So exciting times, John. We're not going to let Amy off the hook here. Amy, a couple questions for you. Let's first start with: do you have a favorite episode of the podcast?
Yeah, I think that's a really tough question. So I'll answer it in a few different ways. I think if I go with my very favorite episode, it would have to be the first one because it's the most memorable for me because I was not sure what direction we were going. And it was, what is this podcast and what are we going to do and how is it going to go? So I just remember, how great it was. And I was just really so proud and impressed. And it just was really good. So the first one is really memorable to me. But then after that, they've all been really great. But I have to say, every week, while I really like the content, the news, I've stopped watching the local news because I find it very depressing, but I listen to the podcast to get your guys' news update because I know I can always hear, the latest and some of the most important things, especially what's going on in the COVID world and get the highlights there. So I love that part of them. And then the other one that really my kids listen in, which I think is quite fascinating to have a couple of teenagers that listen to the podcast here and there. And the one that they resonated with them a lot was, I think it was episode 3, where you really dispelled the myths about antibody testing. And that was in the time of COVID when there was just a lot going on in the news and antibody testing this and antibody testing that. And you guys really tried to put the facts and dispel a lot of the myths in the space. And my kids learned a lot from that episode. So that one stuck with me a lot just because my kids still talk about it. So that's really what I think. But I love all the episodes. I listen all the time. And there hasn't been one that I haven't liked. So I've enjoyed them all.
Phew.
No, I agree. And thanks for the kind words, Amy.
No, thanks very much. But good to know that we meet your approval on this.
Absolutely.
I like that the first one stuck out because you were like, what the? Well, if it makes you feel any better, John and I were kind of under the same impression. No. It's been a joy.
Sit back and laugh too, because you guys are quite funny as well. So there's lots of times I giggle, and there's funny parts to them too. So I find them very entertaining as well. So
funny thing is, we do get edited sometimes, and Jeremy says he might try and get a bloopers going, which would be kind of funny, but maybe not always appropriate with some of the Italian language that comes out of my mouth. No, all kidding aside. So, Amy, before we let you off the hook here, you're on the hot seat, by the way. This is the hot seat. I'm dubbing it. Any guest is on the hot seat because you're going to get asked episodes. So for anybody else in the organization listening that will be on the podcast, prepare yourself for the hot seat. What are you doing this weekend?
Yeah, that's a really good question. I'm glad I am not in the path of Hurricane Laura. So I am not around that. So I'm not really doing much of anything. I'm going to just enjoy being home this weekend and not going anywhere, staying away from people, and just enjoying being home.
Well, good. And everything is the fam home. I know you're humble about this. Your daughter's like a world-class tennis player, at least a collegiate tennis player. Is she playing tennis, or what's going on there?
She is. She attends the University of Mississippi, and they are back in school and back practicing. Still a bit unsure of what their season may may look like, but she's back on campus and practicing. So we'll see what happens.
How are classes and things happening there?
Yeah, there are a lot virtual, but she is taking chemistry class, for example, and lab classes are having a revised lab schedule. So she does go to laboratory in person. It's limited to so many students in person, but she does go every other week to a lab class, and then the off class is virtual. But there are
how come I got a feeling we're not going to see a spike in chemistry lab students, and there won't be a log jam in the chemistry lab with social distancing?
Probably not.
The chemistry students love social distancing.
Like, the chemistry students are going to get pissed if they start having people sign up. Like, what's going on? I know I'd be mad. Like if suddenly, now that I think about it, if I was a biology pre-med student and suddenly people, I just wanted to get some interaction, I'd be like, this sucks. Get out of here, right? I don't know about you. Let's see. The weekend is Thursday. It's a thirsty Thursday. It's been a good week for me. It's been a good week overall. And that translates into things both personal and professional. I got a lot of things done. I think my deck will be done, so I'll be shopping for some outdoor furniture and a mat for a grill. Crazy. I don't know if there's enough time on Saturday for all that stuff. No, I'm just kidding, John. I don't really have anything going on. How about you?
Saturday's a huge day for me. It's record store day. So it means it usually happens in April, and I've done record store days here in Ithaca, done them in London, done them in Raleigh. So it's obviously back to Ithaca this time because I'm not going anywhere. But it's going to be usually it's like if you're really obsessed with music, then you start queuing it, 6-7 o'clock in the morning to get in there early to get the rare vinyl, etc. This year I'm still planning to be there early, but the record store that I'm going to, they're moving it all outside, so it's going to be on the Ithaca Commons, and I'll see how that all works out.
Well, I hope for good weather. That sounds awesome, John. That's some normalcy and sounds really cool.
I have my list ready, what of the ones I definitely want to get, and then there's others that I might pick up because they sound interesting, and then there's some other UK only releases that I'll pick up in the evening as well. So yeah, big day. I got a
feeling you don't have any Tool on there or Iron Maiden or anything like that.
Not this year. No, not last year either. No,
I'm sure it's some good stuff.
three top ones are two bands from the 4AD record label, which was quite a culty label, particularly in the 80s and 90s. And then there's a guy called Terry Edwards who was in a band called the Higsons, where the singer then became a writer and all sorts of stuff. And they're from my wife's hometown, and that's my UK only release that I really want to get. But there's a bunch of other stuff.
Actually, on a slightly different note, my son told me today, earlier in the week, he wanted to maybe work at the comic book store, which was an interesting choice, seeing how that came out a little bit out of left field, given his, a football player and really been doing a lot of athletic stuff. And at one point he was talking about working at the pizza shop and maybe Dairy Queen or all the places that seem a little more what we had thought, but what a cool moment for me. He said he wanted to start thinking about that, and so he triggered me to think about going down to the local comic book store, which we used to do all the time as a kid with him. So that was kind of cool. I think golf and tennis are still practicing, or still high school wise, that is. The football has stopped, which is weird. We were doing practice for outdoors and really doing some good social distancing thing, but they've stopped this week. They're in limbo. Literally, I think they had practice last Thursday, and then it's been up in the air. Like I mentioned before, I think some local schools within the county have canceled all sports for the fall, but here we're still waiting our fate. There'll be no practice this week, and I think the outcome is somewhat pessimistic for football, but we'll see what happens. But I do think things like golf and tennis, they're planning on having those. Although my neighbor last night gave me more hope. She seems to think both Blue Valley and Olathe here locally, Overland Park and Olathe, are going to play. So anyhow, would hate to see him miss a freshman year, but also understand the safety around it.
Sure. I have one follow-up from last week. So in terms of my oven, we actually went and ordered a couple of ovens to replace the one that's going out, and tomorrow I'll be measured up for the space to see how it can be installed, and we're looking at a delivery date of 11th September. So that's going to be good to have a fully working kitchen again.
I now have a dual oven that works. I would be crippled if someone took one of them away. So You'll love it, John. It's great. Cool stuff. I think that's a wrap, right, John? We'll have a great jam-packed podcast number 18. Thank you so much to Amy Mize.
Yeah, thanks very much, Amy. This would definitely not have been the level of podcast it was without you. That was really, really excellent. Thank you.
Well, John, anybody on will raise the level with you and I. And just have a great rest of your week, everybody.